My goal today is to try to finish up the highlights or high spots of chapter 7, and we'll pick up from sort of where we left off talking about pollination in different types of flowers. And the one that's listed in the textbook on figure 722 is a good one for you to use to study by. I've adopted Tom's philosophy of suggesting to you various things that you need to be aware of for our academic exercises, and the one we got coming up is more than likely going to have a graphic of a flower of this type that you will need to identify some components.
Are you trying to show it on the screen?
No, I wasn't trying to. Am I?
Oh, I didn't know if you were or not because we weren't saying it if you were.
No, I didn't put it up, but it's in the textbook. Yes, ma'am.
Is it on page 129?
Oh, I don't know. I don't have a book open. I'm just using PowerPoint. But it's figure 7-22.
7-22. Well, that
Yes.
means it's in chapter 7.
It's right there. It's on, yes. 120.
Okay. Yeah. And it's got the components that you need to be aware of with the petal, and you know what a flower petal is, but you need to be able to identify it. And then it talks about the stigma, and that's that center portion in the flower that is the receptor for pollen. The anther is the yellow portion that contains the pollen. It's on the top of what we call the filament. The filament is nothing but a little string-like structure that supports the anther. And then that style is the tube component of the stigma. And then we've got the sepal at the bottom portion there. And that's basically a little leaf or a little green structure that did encompass this bud as it was beginning to, as it was in its growth stage. And as the flower begins to emerge, that sepal opens and separates and pulls downward. It's a protective device. Then we've got the ovule, which is in the center portion there at the base of the stigma. And that's where you have your ovary. And the ovule is actually the seed, the center portion of the seed, and then that ovary is a component around that. So just make yourself aware of these different structures. Now there's some differences in the flowers that they've got depicted in there. And this one, the way it is done is a depiction of either a peach, a cherry, or a plum, where we've got a center ovule, center seed. Now an apple or a pear is going to be a little different from this because it's got carpels and multiple seeds in it. But as long as you're familiar with these structures, you'll be able to identify them. And you know, I'm not trying to harp on this a lot, but in an introductory plant class, you need to be able to recognize these things. So that's all I'm getting at. And that's as we talk about pollination, there are a certain number of plants that are considered to be self-fruitful. And by self-fruitful, it means that it will be pollinated by its own pollen without cross-pollination. So there are certain plants that are self-fruitful. Most peaches are self-fruitful. They do not require a cross-pollination. Now apples, on the other hand, are generally require a cross-pollination. Your squash, watermelon, cantaloupe, things of that type, they've got a different flower structure, and they've got a male component and a female component on the plant, which requires cross-pollination. It means that an insect or bee or something has to carry pollen from that male component of the flower over to the female flower to be able to produce any sort of fruit. So there's quite a difference there. And as we talk about self-sterile plants, a lot of them, the pollen is just not viable on those sterile plants. So it requires a cross-pollination. There, an apple variety such as a stamen, it is sterile. It will not produce fruit with its own pollen. So it's got to have pollination from another apple variety. Golden Delicious is basically self-fruitful. That's why if you ever had a Golden Delicious tree in your yard, you're going to get an enormous crop one year, small apples. The next year you won't get much of a crop because the plant expends all of its energy on that one year, and it doesn't have any reserves to set fruit butts for the next year. So that's why we get in an alternate bearing situation with some apple varieties because of that fact. That requires a commercial apple grower to reduce the numbers of fruits on a given tree so he'll get size and ensure that he has a crop next year. You know, from an apple grower's perspective, he needs to have a crop, a reasonable amount of fruit every year rather than have a high crop one year, none the next, another high crop. It's hard to manage an operation if it's done that way. Like I said, the pollen transfer is generally done by insects on some of these things. And some things, like we talked about sweet corn, the majority of the pollination of sweet corn is done by wind and or gravity because of the way the pollen is initiated on the male flowers in the top of the plant, and it just drops down to the female. But the silks that occur. In the textbook, it tells you that other pollinating agents are water, snails, slugs, birds, and bats. And to be honest, I've never seen anything pollinated by a snail or a slug. But, you know, the book does include that. So that's just an outlier in my eyes. Skipping over a little bit of this stuff. In a way of definition, the term parthenocarpy is the formation of fruit without the stimulation of pollination and fertilization. Without fertilization, no seeds are produced, and thus parthenocarpic fruit are seedless. Now we get some seedless fruits, like watermelon, as an example. They may be cross-pollinated, but then the seeds in seedless watermelon varieties abort. So that when you cut a watermelon, a seedless watermelon, quite often you'll see little white seed-like looking structures in the interior flesh of the watermelon. Those are actually were the potential for seeds, but in a seedless variety, those seeds abort. Mother Nature has taken those out. So that's one of the key things. At the market that I have, it's quite often I'll get somebody, we'll put out a display of seedless watermelons, and some of these folks will come in and buy one, and they'll come back in 3 or 4 days, and, well, that seedless watermelon I got had seeds in it. I said, really? What do you mean had seeds? Well, they were little white things in it that, you know, were seeds. I said, no, they would have been seeds had they grown, but they were not seeds. Well, it said seedless. I didn't expect to have any of those in there. And sort of gives me an idea, well, these people are not very well grounded, but that's beside the point. We have seedless grapes and seedless, you know, a lot of times the seedless grapes are an example of those that the seeds that were there just abort, and no viable seeds are produced. Seedless grapes are much more desirable than the seeded varieties. And if you're buying them, seedless grape, usually 20, 30, maybe 40% higher than a seeded grape. Seeded grapes are much less costly to produce. So, skipping along a little bit, It talks about fruit set, and I inferred to that a little bit, talking about the apple production. Fruit growers, apples, peach, pear, plum, those type folks need to have a fairly uniform or level of production to be economically viable, and they generally have to manipulate their crop for not only the quality of the crop, but the longevity of the crop. If they allow, say, a variety like Golden Delicious to go unmanipulated, you'll have a huge crop of very small apples one year. The next year you'll have essentially no apples, because of the fruit buds that are initiated for the fruit buds that are going to be for the 2018 crop were initiated last June. And if the apple tree is overcropped, the tree is trying to produce all of the fruit that it currently has on the tree and doesn't initiate fruit buds for the next year. So it's a difference in what we deal with a balance of the fruiting deal. For that reason, the apple growers and peach growers will do something to reduce the level of crop that they have. They have to evaluate how many apples they have or how many peaches. And generally, the apple thinning is done when the fruits are really small, less than the size of a dime, and the majority of that's done chemically anymore, with the application of a variety of growth regulating compounds that will reduce the crop. Peaches, on the other hand, are physically removed. Sometimes the grower will have a device that will actually carry heavy ropes, and they'll drag those across the tops of the trees and just physically rub off a percentage of the peaches. A lot of folks will have a reasonable size crew of Hispanic folks or migrant laborers that will go through with a rubber hose and physically bang the limbs to knock off a percentage of the peaches. So, multitudes of ways of doing that. It gets into some growth curves in here, and I'm not going to go into that with you guys. Another term we talked about very briefly was the one called senescence, and the definition is a terminal, irreversible, deteriorative change in living organisms, leading to the cellular and tissue breakdown and death. So it varies with the type of plant that we're talking about as to the way of what happens with senescence. And if we're talking about an annual, it's going to grow up, fruit, and die, and that plant is gone. If we're talking about a biennial, we're going to have a biennial. You've got two growing seasons, so it generally produces a plant the first year, then it'll overwinter and grow a flower spike or produce flowers in the second year, and then it goes through the senescing process. It begins to break down and go away. Senescence in perennials is quite different because they're more woody, and senescence is usually a process that is quite different with them. So it's a more of an aging process than a type of breakdown. Plant growth regulators are a group of plant hormones, some of them natural and some of them are synthetic. And the synthetic growth regulators are chemicals that are not found in plants. But a plant hormone is a natural substance that acts to control plant activities. And we talked a little bit about those, but as we talk about plant hormones, we come upon the term of apical dominance, and that's the situation where you've got naturally occurring plant hormones that originate in the growing points of plants. And that apical dominance is exhibited by the fact that plants will continue to grow upright until they reach a designated point with the given plant. The example that I like to use with apical dominance is one that is a plant that's commonly grown in all over the south side of Virginia with the tobacco. Tobacco is a plant that will grow up, depending upon the variety, a designated number of leaves. And the plant breeders have adjusted that quite a bit over the last number of years. And back 30 or 40 years ago, tobacco plants grew much taller than they do now. But as a result of genetics, they have improved the quality and basically have a designated number of whorls of leaves that occur on most tobacco plants. But as tobacco grows up, it reaches that point that it's going to begin to flower. And the tobacco growers will go in and take that flower stalk, break it out. The term they use is topping. They'll take that tobacco flower out. Some people are actually doing this chemically now, but the old way to do it was to physically walk through and break out those tops. And when you did that, you removed the growing point and you removed the origins of the plant hormones that were originating this term we talk about with apical dominance. And with the removal of that, you eliminated the hormones. So these hormones had been inhibiting the axillary buds on the tobacco plants. And those axillary buds were in the actual axil of the leaf. Where a leaf comes off from the stalk, there is a bud that originates right there. And when we eliminate that, those hormones in the flower, that enables these axillary buds to begin to grow. And that initiates the problem that tobacco growers have with suckers. So old school, back when I was a kid, you topped the tobacco and about 2 or 3 weeks later, you went back and you physically removed these suckers. Horrible work, hard to do, time consuming, hard, hot, dirty. But a lot of that work is done now. They'll top the tobacco and then they'll put on other growth regulators that will again inhibit. In other words, they'll take the place of the regulator that the plant was originally producing. They'll spray that over the top or pour it down the stalk, and it will actually inhibit the production of those suckers. All of this is done in an effort for the tobacco grower to control the growth of the plants that he's doing. He wants to maximize his leaf area, maximize the growth into those plants to gain as much weight as he can and quality weight for the production of his tobacco plants. So, there's quite a manipulation of plant hormones and plant growth regulators with the production of tobacco. And all of that's a result of the idea that we talk about apical dominance. Skipping over a little bit more here. It talks about gibberellins quite a bit, and gibberellins are nothing but a naturally occurring plant hormone, and they contribute a lot of things to different types of plant production. They're using artificial gibberellins in the production of seedless grapes. And there's a picture here, 732, of a series of grapes that have been treated with gibberellin sprays, and it greatly improves the size and the quality and the overall appearance of these seedless grapes. But it's just an application of a GA, or gibberellic acid, as it talks about. And they've used those a lot in all sorts of plant production. There's a lot of them been used in apple production now. So, and we could have an entire class on growth regulators if we got into it, the plant physiology that's involved. Cytokinins are another naturally occurring plant growth regulator, and quite often in commercial production of different crops, they will use a combination of gibberellins and cytokinins to manipulate various stages of plant growth. But that's all you really need to know about that. The next growth regulator that we need to talk about is one called ethylene. And ethylene is a naturally occurring gas that comes from the ripening of a lot of different fruits, and it's a naturally occurring thing. And the scientists and extension folks have done an awful lot of work during the 1900s, mid 1900s, to gain a better understanding of ethylene. How many of you will have enjoyed a tomato in the last 2 weeks? Commercially produced tomato, not one that's come out of your garden.
I didn't think there's such a thing as enjoying a tomato.
Yeah, Tom. Well, my point being, tomato production during this season until we get into probably June, with the exception of greenhouse tomatoes, a lot of the tomatoes are grown in areas far to our south, and they will be harvested at what we call a mature green stage and shipped north to various wholesalers. And those tomatoes will quite often be treated with ethylene gas to trigger the ripening of those tomatoes. That's just one of the uses of that. Every banana, well, I shouldn't say every banana, but the majority of the bananas that any of you have eaten in the last extended period of time have all been gassed with ethylene gas. You will, bananas are picked in what we call a green mature stage, and they're shipped green. And I've got one wholesaler that I work with that will get a tractor trailer load of green bananas in about once a week. And he will divide those bananas into different lots. So he may take the 1,000 boxes of bananas that come on a tractor trailer and divide it into 250 box lots. And he's got little small rooms that are a little bit smaller than this classroom that we're in. And he's able to regulate the temperature, and he also controls the amount of ethylene gas that's there. But all of your bananas have been gassed with ethylene to initiate the ripening. And it's an interaction between the temperature that they hold the room and the amount of gas that they put in. But this gentleman he's got his recipe down right. He knows how many bananas to put in each room, and he'll set, say the truck comes in on Sunday morning, he divides it into 4 lots of 250. He's totally out of bananas. So the first room, he puts 250 boxes in. He'll say put in 300 parts per million of ethylene gas in that room and turn the temperature up to about 70. Close it up. That's on Sunday morning at 8 o'clock. Monday morning at 6 o'clock, he can open that room and those bananas will be a bright yellow with a green tip, just perfect for selling. The next room, he'll go beside it and he'll put 200 parts per million of ethylene gas in and crank the temperature in at about 50. On Wednesday, he can open that room, his bananas will be just right for selling. He can stage and control the ripening on his bananas by the amount of gas he puts in and the regulation of the temperature. So it's something that's to me is just kind of fascinating. Mm-hmm. The tomatoes we're talking about, quite often, the same guy may order a tractor trailer load of tomatoes coming out of Plant City, Florida. They pick mature green tomatoes, put them in the truck. He's ordered break-in tomatoes, which means that they are pink and beginning to turn red. They put those tomatoes in the truck in Florida. They'll put a gas bomb, basically, it's a gas canister, in there, in that truck, sealed, and turn the temperature up to about 70. In the 12 hours it takes to get from Florida to Winston-Salem, those tomatoes have turned from a green to a pink, ready to sell. They're gassed on the road. Never make, it's part of the science that's involved with the manipulation of the various types of produce that we get. But ethylene gas emerges from ripening apples. The ethylene gas itself has some kind of unique things. The old timers in our area in particular would have a root cellar, and quite often the root cellar would be a basement area that they had that dug back into the hillside, and would, on normal days, temperature in the root cellar is going to be about 55 degrees because of the ground temperature and so forth. But during the summer, they grow a decent volume of potatoes because they weren't used to going to the store and buying 5 pounds of potatoes every time. They'd have 10 or 15 bushels of potatoes that they grew in the garden. They put them back in the root cellar. And in the fall, they would also go get 8 or 10 bushels of apples. Also put those in the root cellar. Apples emit a naturally occurring ethylene gas as they ripen, and the ethylene gas from the apple will inhibit the sprouting of the potato. So it's a symbiotic relationship with the emergence of the gas to curtail the sprouting on the potatoes. The old folks knew exactly what they were doing and how to do it. Fairly constant temperature, so it worked out really well. Ethylene there's a lot of synthetic ethylene being produced, and they're using it to actually ripen tobacco in the field. Assume that you've got a large tobacco grower, and for one reason or another he's gotten a little bit behind with his harvest in the fall. He can spray ethylene, a spray solution of ethylene over a volume of tobacco, and go in, and it'll ripen it in about 4 days. He can go in and harvest that entire field. So, that's a catalytic effect that can occur from the ethylene. It's an interesting chemical. It causes fruit ripening, flower initiation. It can change the sex expression on plants. It can degreen oranges, lemons, and grapefruit. One of the first uses that they found commercially with ethylene was in the production of sour cherries in Michigan. They were having a real problem with the harvesting of cherries, and they physically could not get them harvested by hand, so they were trying to come up with some solutions to that, and they built some machines that were basically a catch frame that they would put underneath a given tree and shake the tree. But the cherries didn't fall as rapidly as they'd like, so they learned that they could spray that tree with ethylene, and then in about 3 days do that shake, and all the cherries would drop down onto their catch. So it's totally changed the harvest. It's a harvest aid. It says it can be harmful to plants, can cause unwanted leaf abscission, and that's basically what we're talking. They said that's a harm for plants, but they've modified the use of that for tobacco to actually use it as a tool. I know when I was in Blacksburg, we did some work on thinning of apples with ethylene, and it was pretty critical. If we sprayed it when the temperature was too hot or the concentration was a little too high, we take all the flowers off. And if we was a little cool or the concentration wasn't enough, we didn't take enough of them off. So it was something we were working with to learn. Skipping over a little more here. A lot of synthetic growth regulators. Most all of you are familiar with a herbicide called 2,4-D. It's an old material, been around since the mid 1900s, and 2,4-D is another growth regulator. It actually causes the plants to grow till they destroy themselves. Most of you guys are too young to remember the crisis that occurred in the apple industry with a term material called ALAR, but there was a group in California that were raising Cain about the use of ALAR as a growth regulator on apples. It was a wonderful tool that we had, but they had found some research that said that one of the breakdown components of ALAR, when heated, would be potentially carcinogenic, and they were raising Cain about the use of ALAR on apples that were going to be used for baby food, for applesauce. So every time I see Meryl Streep, I get mad because she became the paid spokesperson for this group that were raising Cain about the ALAR, and she knew absolutely nothing about what she was talking about. But since she was a recognized movie star and paid spokesperson for that group, she basically was one of the principals that was involved with having ALAR taken off the market. And it was a real detriment to the apple industry because it had some wonderful characteristics of increasing the red color on apples and making them firmer and delaying the maturity. Wonderful news. ALAR is still being used today in the nursery industry. Instead of being called ALAR, it's called B9. And it's a material that is used to reduce the growth on some plants. Chrysanthemums, for example, or the poinsettias, they can put B9 on those, even bedding plants, to reduce the growth and make it more compact. So if you've got a greenhouse full of commercial tomato plants you planned on selling to garden centers, and we happen to come in with 10 days or 2 weeks of cold, damp, wet weather, tomato plants aren't being sold out of your greenhouse like you needed to, you can put an application of B9 on them and prevent them from elongating and getting too big to sell. Just one of the things, the tools that happens with those. There's a bunch of other plant growth regulators that are used as retardants, and I won't expect you all to be familiar with those, but that's just a snapshot of some of that stuff. And that's the last slide that, excuse me, that is in Chapter 7. So, any questions? I've gone through that very hurriedly, but tried to hit the high spots on things that I thought might be of value to you. So,
I have a question,
please.
Mr. Ayers. So I was looking in my Blackboard stuff, and there's a couple, I don't know if you've gotten to like everybody's lab reports. I know I submitted all of them.
I haven't printed and graded any of those for the last 2 weeks.
Okay, all right.
I've been fighting other fires, but after we get through class today, I'll have a little opportunity. I won't get it done today because I've got too many other missions, but I'll get those done this week. Now, please help me by the fact of, if I don't get your grades for the lab reports in this week, send me an email and let me know. But there's a little bit of confusion on my part on some of the lab reports you guys have done compared to what we've done here. So,
me too. Yeah.
All will be fine. Don't worry.
Okay. Thank you.
Yeah. Any other questions?
What chapters are we covering on your test on?
It will be through Chapter 7. What did we go through? 5 on the others, so it'll be 6 and 7, I think.
5, 6, 7.
Okay.
Oh. And then.
So, was chapter 5 on first test, or just
I think he was on the first test, so it's 6 and 7.
6 and 7? Okay, just the way Chief said.
There's a lot of stuff in these 2 chapters.
Yeah.
So there's enough material to.
So chapter 6 and 7 this Thursday, and then all 1 through 7 next Thursday.
Right.
Okay.
I printed out your
Study guide.
study guide for the final exam. So, is there a study guide for this,
Yes.
the one on Thursday?
No, I don't think so.
Alright,
But if you've got the study guide for the final exam, if you can find out the
go through that.
break off for where chapters
Yeah.
6 and 7 are,
I'll
then
do that.
there you go.
That's really the reason we didn't do it this one.
That makes sense. Alright, no worries, thank you.
And if you've got questions, send me an email. I'll try to get back to you.
Thank you, Mr. Ayers.
You're welcome.
You're very welcome. You're very welcome.
Y'all,
Anybody in Keithville have any questions?
I forget, they're even here.
Yeah, I know.
They're so quiet, I don't know whether they even hear part of the time or not. What do we do with Keith?
I
We were just
forgot.
saying, we're like,
Well, the stress may be getting to Shannara so much that she just is at a loss for words.
It's okay, Shamara.
Shush.
I wish I had the number of folks that you got, Tom, between the 2 places.
Oh, between the 2 classes, I got a dozen people, probably.
I know, and that makes it a lot easier. I've been in here talking to an empty room all morning, and I don't like that, but that's okay.
Yes, I've done that before myself, so. But
As I said before, if you've got any questions about the test for Thursday or the final, don't hesitate to send me an email. I'm running around all over the place all the time, but the emails will bounce in my phone, and I can stop and try to help you. So.
Okay, sounds good.
yeah, and John, I just sent you the animal final, so.
Okay, that'll be fine. And Tom, I don't know exactly. We're going to basically follow our exam schedule, which is greatly different than yours.
Yes.
So, if I don't do that, I get into all sorts of conflicts with the other classes that our folks have got. So.
Oh yeah, that's what I'm figuring. You do your thing, I do my thing. That's why I was trying to say if we all have all of our tests made before finals week ends, then we all are covered. Which I'm going to hopefully have my 231 done and sent to you this afternoon.
Well, I think I sent you the 142 and the 234 this morning. So.
Yep.
But if